One of the critical areas, though, that I want to mention here is the impact this had at this time, all this uncertainty around the scope as the guideline in terms of how it applied to existing products. What did the guideline, the European guideline, which was the issued guideline, say? Well, it included quite a lengthy statement, which is reproduced here. What I'm going to do is break this down, but there are several areas within this I've highlighted. I'm not going to read this out in its entirety. I'm going to pick different parts of this now and note what it actually means in practice. The one I want to start off with, though, is by mentioning the fact that it starts off by saying it only applies to new active substances. So it begs this question, well, what about an existing product that was developed before 2007 when the guideline came into effect? So how do you interpret it? Well, as I said, there are several parts of the key in this. The first one is this. It says it also relates to new applications for existing active substances where assessment of the route of the synthesis, process control, and impurity profile does not provide reasonable assurance that no new or high levels of genotoxic impurities are introduced as compared to the products currently authorized in the European Union that contain the same active substance. What does that mean? Well, it could be interpreted to mean that to introduce a new version of that API, that what you would have to do as a minimum is prove that the levels of any genotoxic impurities within it were no higher than those present in the currently authorized product. Sounds simple, but what about in practice? Can you do it in practice? Well, actually no, it's extremely difficult to do this. How can you compare the profiles, particularly when the initial profile is not disclosed? In fact, if the initial profile may not even be known, for instance, if it's a very old drug and there's no changes being made to it, guideline doesn't apply, why would anybody have done a mutagenic impurity risk assessment on it? So we may not even be able to do that comparison. And you could be even comparing two different processes. And so from both the applicant's point of view and the regulator's point of view, this is a well-nigh impossible task. It also goes on to say the same also applies to variations to the existing marketing authorizations pertaining to the synthesis. So what does that mean? Well, this is perhaps the only bit that is quite straightforward. If you make a change to synthesis, you need to assess the effect on the levels and the nature and actually what genotoxic impurities might be present. So that's fairly straightforward. But the real killer in this is the next statement. The guideline does, however, not need to be applied retrospectively to authorized products unless there is a cause for concern. I talked about nuances in English earlier on. This is a very bit of nuanced English from the point of view of cause for concern is a very vague term. What does this mean? It was initially very unclear. Was it a structural concern or was it actual safety data? So, because of this, this was again one of the areas that was subject to that Q&A process. And this particular question that's highlighted here was posed. The guideline does not need to be applied retrospectively to authorized products unless there is a specific cause for concern. What might constitute a cause for concern in terms of application to currently products? So basic question, what do you mean by cause for concern? And this was the response. And if you ever wanted an example of vague English, then this is the go-to example. Just read through it. If a manufacturing procedure for an API remains essentially unchanged, a re-evaluation with respect to the presence of potentially genotoxic impurities is generally not needed. However, new knowledge may indicate a previously unknown cause for concern. Clear? No, not really. Cause for concern is not clarified, and the example, certainly of sulfonate esters, is probably a very poor choice actually, because the reality around whether or not that is a concern is highly debatable. So that didn't really move us forward. Interestingly, at a similar sort of time, in parallel to this, and I'm not clear what the actual trigger was, but there was an EDQM article, this is within Pharma Europa, so this relates to the European Pharmacopoeia, in 2008, where they started to define what their policy was for dealing with genotoxic impurities when you're talking about developing a monograph. And this is generally helpful, but one of the things that was really important in it was it went as far as making a statement which to some extent helped clear up cause for concern. It said that action is needed only where there is study data demonstrating genotoxicity of the impurity. The existence of structural alerts alone is considered insufficient to trigger follow-up measures. This is of course relating to an impurity in an existing product. And it also went on, and provided this very helpful appendix showing a series of scenarios